In this video, we will discuss about some of the molecular etiology of CAR T cells related cognitive impairment in the patients. So basically, we want to find out the origin of this cognitive impairment, or the researchers focused on finding out that. So they took the patient samples, and this is how the workflow of the experiment works. In the panel A, it has been shown they took the patient samples, did the nuclei dissociations, then sort the cells using this DAPI fluorescence, and thereafter did this 10x sequencing, and then use this single cell nuclear sequencing analysis to find out whether there is what is the origin of this CAR T cells related cognitive impairment in the patients. So once first they plot this uh, single cell nuclei seq and data in different populations based on their markers, and they have distinguished these populations in different categories that is mentioned here, and all of them forms a cluster based on the markers that are present in those uh, nuclei or on these samples that is shown here. And most of them are this neuron, and some of them are the microglia, astrocytes, oligodendrocytes, and other that are shown here. Thereafter they use this microglia and in cluster them based on the patients, and they are able to see that for each patients along with this control forms a separate cluster, and we can see that the macroglia clusters and the patient clusters are almost similar to each other. And then they wanted to find out this how much is the changes that has been observed, how much the changes in the gene expressions are been observed in the patients compared to the control. And we can see that the changes are very high in the CAR T cells treated compared to the controls. And then they want to find out what are these changes, and they can see that in different patient samples uh, different changes occurs, and there is no uniformity. Compared to the control, we can see that for this MGA patients, we can see the upregulations of some of these pathways which are not upregulated in the case of controls. And the main observations of this panel G is that different patients behave differently in terms of the change in gene expressions, because in the cancer cells the mutations occurs differently in different patients. And thereafter they check some of the important parameters using this violin plot, and they are able to see that these two parameters that are upregulated in most of the samples is this CX3 CR1 and P2RY12. It's upregulated in almost all the samples except this MGD that can be seen here. And then they wanted to delete the gene set enrichment analysis to find out what are the genes that are enriched in different patient samples, and they are able to see that the different patient samples has different sets of genes that are enriched. For example, MGA, this enrichment is of TNF alpha IL1, INF gamma IN1 is seen, whereas in the MGB they see the upregulation is mainly in this region that seems to like MMS, whereas MGA it's mainly about TNF, and so MGA is mainly about TNF. So, and this green color indicates the cytokines that induce polarization state. And for the MGB, they see the most upregulation is MMS, and for the MC1, the IL1, and the MC2, MGC2 is also the IL1, but for the MGD, they see the IL-1 and inter IFN gamma are the genes that are highly upregulated or dysregulated, we can say that. Then they, after validating, checking their patient samples directly using the single cell nucleic analysis, they also check the cell lineage of these patients. And when they cluster them, when they cluster this oligodendrocyte lineage, they are able to see the different kind of lineage, and this is also in line with the different patients. And this indicates that all these patients, in all the patients, the effect of CAR T cells is different, and also effect of CAR T cells is different at molecular levels because of the uh, because each patient has different kind of mutations. Then they check the variations in this OPC and oligodendrocytes, and they can see the variations is high compared to the control in both the cases, for the oligodendrocytes and also for the OPCs. That proportions of the patients that have these OPCs are high in the CAR T cell treated, whereas it's very low in the case of control. And similar results for the oligodendrocytes, that the patients who are treated with CAR T cells has high oligodendrocytes, except for this patient A, that can be seen here. And here in the panel D also we have seen these similar results, that variations are high in the CAR T cell treated patients compared to the control. Then they also did the cell lineage, this heat map analysis, and they are able to see the similar things, that each patient has different transcriptions genes that are upregulated or dysregulated, and they checked it for these OPC clusters and also for these oligodendrocytes clusters, OLG clusters. So all these results overall indicates that there are changes at the molecular etiologies, and so far they are not able to find the common origins, but they can see the changes are there in the patients who are affected with patients who are treated with CAR T cells, and each patient behaves on its own. That has been seen in these results. So further studies needs to be developed or need to be done to find out specific patterns in different kind of patients. That is what is mentioned in the paper also. If you need to understand more about this molecular etiology of CAR T cells, related cognitive impairment in patients, how they did their studies and how they did their bioinformatic analysis, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.